Well, thank you so much. I'm just in love with this meeting and the concept. I've tried for many years to pull the land of cell therapies out of the cancer clinic, and I'm just so excited to see everybody in the same room that I've looked up to for so many years. So I'm excited to tell you a little bit of interweaving set of stories that combine two different things that are kind of new to the day so far. The first is cardiac fibrosis instead of a B cell target, and the second is the method of generation of these cells. We heard earlier that CAR-Ts, as you pull them out of the body and virally manipulate, are very expensive. So we've also developed an approach that I'm excited to share with you that pretty much bypasses all of those things. So I don't have any financial disclosures. I'm not lucky enough, but other people do on the team, especially the privately funded company called Capstone out of San Diego. So I'm going to simplify fibrosis. We're in the context of the heart most of the time. It's very complicated, but for the sake of argument, there's a homeostatic fibroblast or pool of fibroblasts that are making your extracellular matrix, the scaffold in which heart muscle cells sit in and pull against. And in the setting of disease or injury, they can activate and change both molecular and phenotypic characterizations. And the real pathogenesis comes from these activated fibroblasts that are molecularly distinct from these homeostatic fibroblasts. This is a vast oversimplification, but it demonstrates the point well that a lot of the therapeutics are aimed at really kind of blocking the transition between this state, and they don't really work with 100% efficiency. So we thought, well, maybe we could eliminate the activated fibroblasts specifically with a CAR-like protein. So what do you go after? What's our target? What's our CD19 of the activated fibroblast world? Well, we first looked at a bunch of injured hearts and just compared these two different long-term cardiac diseases compared to their non-failing counterparts. And the top protein that stands out is upregulated in these activated fibroblasts as a protein, creatively called fibroblast activation protein, or FAP, which is finally a protein that matches its expression pattern. So we designed a CAR-T along with Steve Albelda and Ellen Pure at the University of Pennsylvania that will recognize this fibroblast activation protein along with co-stim domains. To show you that it's expressed, this is work from Pedro and many, many others throughout the years. We're looking in green at the expression of FAP. It's not found in non-failing hearts or healthy hearts, but it's really robust in all manner of different cardiac disorders, both acute and chronic. For the settings of this talk, I'll talk mostly about chronic models of heart disease. And what's really cool is a group here with Frank Bengal who've developed a FAPI PET probe, so we can visualize in humans and animal models the expression of FAP after MI and other diseases, both in humans. And high expression of activated fibroblasts corresponds and correlates to a long-term loss of function or lack of recovery. So that really high burden of activated fibroblasts is contributing to a loss of function over time. So Halle Agajanian here, who is the postdoc in John's lab who initiated the anti-fibrotic CAR-T projects, is now at Capstan, really kind of brought these two technologies together, the fibrosis angle and the CAR-Ts. In a mouse model of hypertensive injury, we drive with angiotensin phenylephrine. You can see in pink here, all the fibrosis. This is just a histologic stain. So everything that's fibrotic, that extracellular matrix, is accumulating with injury. And with doses of ex vivo virally prepared CAR-Ts, we see clearance of the fibrosis and improvement of the function. So this is wonderful. We thought this was really exciting, really can stimulate a lot of good thoughts, but we know that activated fibroblasts are really important in the immediate injury settings. If you have a heart attack and your heart cells are dying, you need fibroblasts to activate and patch that and hold the ventricles together. So we don't want the setting of a chronic heart disease patient to have persistent anti-fibroblast CAR-Ts. We needed something transient. So we went down the hall and talked with Drew Weissman and his now independent faculty, Hamidi Parhez, to work on a transient approach to deplete these activated fibroblasts specifically and leave those homeostatic, healthy fibroblasts alone. So what we came up with is essentially a vaccine. We have RNA here encoding the anti-FAP CAR. wrapped up in a lipid nanoparticle. This is just like a vaccine for COVID-19, but what we have to do is we have to trick the T cells to take up that vaccine to express the CAR. And to do that, we decorate the outside of the lipid nanoparticle with CD5 antibodies, where CD5 is a receptor normally expressed across a lot of classes of T cells. That internalization results in CAR expression. So first, I'm just going to show you a relatively sparing amount of data, and happy to answer questions further on, but essentially to show you that they can generate, after a single vaccine-like dose, these active CAR T cells in the body. We measure at 24 and further time points after injection intravenously by flow cytometry. And a lot of the core contributors are Tyler, Amir, and Istvan in terms of the RNA production and LMP. So what we're looking at is live splenocytes from these animals that were intravenously injected. And we have CD3, so this is marking all T cells, and then FAP CAR. And as I reveal, what we see is really strong expression only in the T cell compartment, strongly suggesting that this is specific for T cells and not just randomly transducing splenocytes. By 7 days, it's really completely gone, so we've got that transient nature that we want for the anti-fibroblast activity. So what about the heart? We injure the heart, what happens? It's cool, we can make CAR Ts in vivo, but are they actually doing anything productive to help the heart? And indeed, the answer is very strongly yes. So what we have is we're measuring by echocardiography, so an ultrasound picture of the moving heart in systolic or contractile function. In these gray bars, we have the healthy controls. In yellow, the uninjured. Also corresponding, the non-targeted lipid nanoparticles look just like this. But again, a single dose given 2 weeks to recover shows really back to wild type performance of the systolic and diastolic function. And the histologic fibrosis looks very similar in terms of trend. So as I wrap this first vignette and move on to some others, we think it's really exciting that we can have a mass manufactured product. It's an RNA vaccine, like we've been able to dose billions of people with so far. And it's a universal product. Anyone that needs this therapy can get the same exact dose. And if you need a repeated therapy in a few weeks, months, years, you can get the same product, hopefully at a significantly better access and lower cost than current CAR T cell therapies. So one of the odd observations I made really early on, and what we've kind of discussed is in terms of autoimmunity built against the CAR itself, flying under the radar and inhibiting CAR T function. What I identified is in these animals that were treated with anti-FAP CAR T cells, the antigen from the heart shows up held within the CAR T cells in the spleen. This is a process called trogocytosis, and it was first observed in the late 90s, where T cells were acquiring MHC molecules from APCs that are labeled in GFP. And this has actually been shown in the context of CAR Ts, but it comes from the Greek word to nibble. So trogos means to nibble or to gnaw off of, and to bite. And you can see it visualized here, where it's this even bidirectional sampling of the membrane. So to show you that this happens in the context of an anti-fibroblast CAR T cell, we have target fibroblasts here labeled with a FAP conjugated RFP in magenta. And then the CAR Ts have been added, and I'm taking pictures on a confocal microscope, and you'll see them right here forming an immune synapse, and then pulling off puncta that you can see the synapse and the puncta here. It's a little bit dim, but you can see. This is in vitro. What happens in vivo? When we look at the spleen, we only see FAP showing up in the context of the heart being injured and the CAR T cells being on board. And if you don't have both of those settings, there is no FAP retained. For example, if you injure the heart and give them a bunch of extra autologous T cells, you don't see any of this antigen being retained in the spleen, and vice versa. If you give them CAR T cells specific for FAP, but you don't injure anything in the animal, so there's no FAP being expressed, then there is no FAP being retained. And again, it's really within these white pulp regions or T cell regions. And if you zoom in, now co-staining in yellow for the engineered cells and pink for FAP, the antigen from the injured heart, those activated fibroblasts, you can see in cross-section it's clustered here right at the cell membrane. And why this is really important, and we should all pay attention to this, is because now you have a setting where you have a CAR T cell against FAP, and you have a second CAR T cell against FAP that's holding on to the antigen FAP. So now you have the setup where you have both this process called fratricide, where you can have naive T cells that are targeting the antigen that's displayed on the CAR of the other CAR T. And this initial naive CAR can now kill through that linked antigen the other CAR T cell. And this fratricide means sibling killing. And it's been demonstrated in the context of CAR Ts to really inhibit the amount of expansion or usable CAR Ts that are in circulation. So what I told you at the very, very beginning was that we have a couple of different flavors of activated fibroblasts, and I was oversimplifying it by saying there was a homeostatic and an activated fibroblast. Well, one of the really strong pieces of evidence is that we see an activated fibroblast clustering around the arterioles within the heart only after injury. Histologically, it looks just like this. So this is a mouse heart that's been injured with high blood pressure, then treated with CAR T cells. The interstitial fibrosis is completely gone. But what we have is latent perivascular fibrosis that's completely cellularized. So there are fibroblasts there, and they are important for pathology, and they're not FAP positive. So they're not the classic activated fibroblast phenotype. When we look via immunohistochemistry, we can tell with red, which is a little hard to see, but those are kind of a pan-fibroblast marker that will identify most everything. Then we have FAP in green, which you can see out here and down below, and then in yellow, or in white, sorry, is the those little periarterioles. And you can see that there is really kind of robust proliferation in this area that is FAP negative, again, supportive of what we see after therapy. So via lineage tracing, PGRF beta will pick up a broad swath of fibroblasts, both in the vessel wall and in the interstitium. And those are labeled in red here, thank you. And you can see a pretty sparse labeling, which is exactly what we want. This is an uninjured myocardial slice. And then once we injure, we can see just with light microscopy, the arteriole here, and then really expansion of the fibroblasts population in response to the injury in that perivascular space that is FAP negative. So you can see the green staining of FAP out here in the edge, really reinforcing the idea that this is a unique population of activated injury-responsive fibroblasts that's not being targeted by the FAP CAR T cells. That may very well be histologic or pathologically important, not just a histologic finding. So this was reinforced. This was my work, but then it was reinforced by Raphael Kramen's group here in Germany. And what I think is really exciting, more than the hypothesis, is consistent across continents and across mouse strains and laboratories, which is really fantastic. It really mirrors, in my mind, where we have a bridging of a tissue-based immunity or autoimmunity and the injury reactivity. So what I mean by that is, and this is Pilar Alcaende's group from Tufts, this has really worked, along with many other groups around the world, to ascribe a fibrotic role to T cells. So the T cells can, within the heart, they respond to the injury, but they are also independent drivers of fibrosis. And what's really interesting to me is that these, the T cell driven, so Th1 adoptive transferred T cells, then given a high blood pressure surgery, are critical for forming that perivascular but not interstitial fibrosis. So perhaps there is a really interesting bridging between the endogenous T cell compartment and this perivascular activated flavor of fibroblasts that we're really not picking up with the anti-FAP CAR. And again, just to kind of almost bridge the two ideals again between fibrosis and immunity, there is also a potential role for the activated fibroblast to serve as an APC in that context of the perivascular fibrosis, again reinforcing that there may be some really interesting pathology here that we haven't yet explored. So you're probably thinking to yourself, the heart is great, it's injured, fine, but my favorite organ you haven't talked about yet, and it probably also gets fibrotic, and that is absolutely true. And the mouse models are kind of all over the board, and I don't understand most of them. I frankly struggle to keep up with the heart, as complicated as it is. But what we have done is we've looked for FAP expression on a lot of different diseases, and we've started to make some progress with a lot of different collaborators in terms of seeing whether this application of anti-fibrotic CAR-Ts may work in other diseases outside of the heart. And I'm just going to show you one quick example with Adam Cross Group at the University of Birmingham that may excite you in a different direction, and again, try to bridge the gap between fibrosis being a stiff matrix and fibrosis as being an active immune component. Because we often think of rheumatoid arthritis as an autoimmune disorder, and it very much is, but the activated fibroblasts in the joint are a strong player in that pathology. So Adam set up a lot of work, he's now independent, in showing that FAP is expressed in these synovial activated fibroblasts. They drive the inflammation and the pathology and the bone destruction in this area, and they come in a couple of different important flavors. And using a genetic model of depletion, they can show really strong improvement in disease outcomes in these two different animal models of rheumatoid arthritis. And I'm going to move on here a little bit quickly just for the sake of time and show what Sam Kemble, his postdoc in Adam's lab, has been able to show through the pandemic and since that when you give anti-FAP CAR T cells to these rheumatoid arthritis model animals, you get a significant reduction in joint thickness, which is a readout of disease severity. So this first flow cytometry is a little bit complicated, but essentially it means that these CAR Ts are getting into the joint only when the joint is injured. So when there's antigen, the FAP, then the CAR Ts have impetus to immigrate into the joint. The number of FAP-positive fibroblasts by flow cytometry goes down as expected when you get active killing. And what's really exciting is that the joint thickness is decreased and consistent across cohorts. And that's just a little bit of a teaser in one model outside of the heart for the anti-fibrotic CAR T. And normally in my last two slides, I really like to say, well, the CARs are really exciting outside of cancer, but this wonderful meeting demonstrates that I don't need to belabor that point. And instead, I will say RNA is really cool. I show you one method, but there are many, many other different options. I've just put a little box around all these different things. These are very, very easy to manipulate, and you can completely change the outcome of your therapy. For example, you don't need to target effector T cells. By switching out the antibody here to target a different class of T cells, you can make regulatory T cells entirely within the body, potentially for acute cardiac diseases, and a story that I don't have time to share. So with that, I hope to just stimulate your thinking a little bit in terms of how it might be applicable to your favorite disease. I think that the marriage of disease and technology together is very important. And as everyone before me has said, this does not happen alone. This takes an enormous village. And I'm eternally indebted to John Epstein at Penn, who just got promoted to dean and therefore has very little time for symposia that he would very much like to be at. And he sends his greetings. Lastly, I have to thank my current postdoc mentor, Marcus Bugert, at the Center for Infectious Medicine, our funding sources, especially Drew and Hamid's labs, and all of our collaborators. And with that, I would be happy to answer any questions.